molecules in the presence of water to inorganic phosphate molecules, and the enzyme is known as pyrophosphatase. The fourth class of enzyme are all lyases, which are able to cleave C bond, CO bond, and carbon nitrogen bond. But interestingly, they are different from the previous class in a way that they leave behind double bond structure, or maybe they leave behind a ring, or maybe they add a group to a place where the double bond exists. So, in a way, one or the other way, double bond is involved in the reaction catalyzed by these enzymes. A systemic name given to this class of enzyme is a substrate group lyase, beginning with the name of substrate, and then that is followed by name of the group, and finally the term lyase. Now, hyphen is always needed to separate the term group and lyase. So, it is better to say hydro-lyase rather than hydrolase, because otherwise the confusion would arise and this could be avoided. So, therefore, IUPAC has recommended the dot and the dash must be inserted between group and the term, that is standard ending of lyase. A common name for lyase type of enzyme are very simple. You can say decarboxylase or aldolase or dehydratase, etc. In many instances where reactions are reversible also, then the enzyme could be carrying out lyase activity or maybe a kind of synthetic activity if the reaction is proceeding in the reverse direction. So, in that case, synthase is used, but not synthetase, because synthetase is to be used in case of joining two different molecules using the energy by ATP. So, that makes the important distinction between two terms of synthase and synthetase. Now, the third and last digit, the second digit for lyase type of enzyme could be 1 if it is carbon-carbon lyase, it could be 2 if it is carbon and oxygen lyase, whereas the third number in the EC nomenclature for lyase depend upon the kind of group which is being eliminated. If carbon dioxide is to be removed, then it is 4.1.2, 1.1. If water is removed, then it is 4.2.1, etc. So, following the list which is prepared by EC Commission, the second and third digit numbers are accordingly given by the Commission. The another class is of isomerase. Now, there are varieties of isomeric forms which are geometric and structural type of isomers, and any sort of structural or geometric changes being brought about by enzyme are in fact known as isomerization, and it comes within the purview of isomerase. Different type of isomeric activities and isomeric enzymes are there. Some of them carrying out isomeric conversion, some are carrying out racemic conversion, they are known as racemases. There are epimerases which converts between epimers, and there are cis-trans isomers which are able to convert cis to trans or trans to cis type of patterns. There are simple isomerases, tautomerases which are able to carry out tautomeric shift in the molecule. There are mutases and cycloisomerases which carrying out mutarotation of the compound, maybe cyclization of a compound. So, in a way, this type of enzymes are not carrying out C change in the molecule, but they are simply converting one of the isomer into another isomeric form. Simply, this type of catalytic reactions are known as isomerization, and that could be of pertaining to any kind of geometric or structural changes within the molecule. But the empirical formula doesn't change. That is the main consideration. Now, this conversion could be intramolecular also, and this intramolecular oxidation reduction can also take place, but despite that, it is not classified as oxidoreductase enzyme, because the overall net difference within the whole molecule is not significant to place them, place the conversion into either oxidation or reaction distinctively. The third type of group, third level of classification with isomerase type of enzyme is determined on the basis of substrate which is used by the enzyme. So depending upon substrate, third digit is derived, and fourth digit is a distinctive identity of an enzyme which makes the enzyme unique. And the last class of enzyme are all ligases, which are able to join two molecules together, and this coupling of the two molecule is involving hydrolysis of diphosphate bond in the ATP, or maybe any other forms of energy rich compound. Any type of nucleoside triphosphate derivative could be involved to donate the energy. Now, systemic name in case of ligases is derived from a pattern, that is X colon Y and ligase into bracket ADP forming, because requirement for ADP, that is formation of ADP, is essential part to ligation process. So it has to be shown in bracket wherever the enzyme name is given. You can look into the example which is given below. The enzyme commonly known as glutamine synthetase, as described in the reaction catalyzed by this enzyme is described in the left, wherein you can see that glutamic acid converts into glutamine by an enzyme called glutamine synthetase, and the process requires involvement of ATP and insertion of ammonia molecule. So it is merely an extension of a small chain of glutamic acid into glutamine by adding extra molecules of ammonia. So that way a process is of a ligation, and the common name is glutamine synthetase, but a systemic name could be glutamate-ammonia-ligase, and finally adenyl transferase. So this makes the complete systemic name of the enzyme on the basis of X semicolon Y ligase pattern. So glutamate is one of the substrate, ammonia is another substrates, and ligase is the type of reaction which is catalyzed. Now the term synthetase was used commonly, has now been abandoned in common names. It has been suggested by the Commission that synthetase has been used in past in a disorganized way. That is to say, without distinctively recognize the role of involvement, role of ATP in the enzyme catalyzed process. So the Commission has recommended not to use synthetase at least in the common names. Now, second figure in case of ligases kind of enzyme is in fact could be one where carbon oxygen bonds are involved, could be two if carbon and sulfhydryl bonds are involved, as in acetyl CoA, etc. So depending upon the guideline given in the EC list, the second number is derived. The subclasses are only in carbon nitrogen ligases. Now in the next slide I would going to request Rikin sir to tell you about typical classes of each one of these enzymes, how they are listed in the Commission, how many entries are there in the EC database. In fact, you need not to remember everything about this database information, because this is just to give you some idea about how the information are organized. We have tried to put a sample before you, that if you refer into the enzyme database structure or EC database structure, this is the way you are going to retrieve the information. So having a look at the slide is going to give you some idea about the naming pattern which is in practice presently for different classes. So I would request Rikin sir to tell you something about these classes in detail.
So As Yagnik sir has discussed in greater detail about naming and classification pattern which is given by Enzyme Commission, that what type of exact rules to be followed, and as the example we have taken, that is very famous one, that is EC 1.1.1.1, then what all one means for or stands for, and likewise any type of the number you just think means it is there for some enzyme, then you will be able to understand if you go through the all different legal rules, or you can say the rules and regulations which is given by Enzyme Commission, then you will be able to understand and retrieve the information regarding the exact enzyme, and the enzyme as well as the reaction which has been carried out, as well as the substrate on which it will be acted upon. So here we are now trying to list out some different enzymes, the name of the enzymes under the specific classes, as we have discussed and understand now that there are 6 basic classes. So as it is very clearly now known that EC1, we are taking examples of first class, that is Oxidoreductases. So EC1 denotes for all oxidoreductases. So subclass, if it is EC11, then it will be acting on the CHOH group of donors. It means CHOH group, if it is acted upon, then the name should be EC1.1. Then 2, it is aldehyde or oxo group, and so on. So different types of examples we have tried to listed upon. I think it is not possible to read out or to explain out all these names here because of the limits of time, but 1, 2 or 3 we will try to discuss you for any for all classes. As we now understand the second class, that is of all transferases, the basic understanding is clear in our mind that in this group whatever the enzymes will be listed will be doing one thing in common, that is they are going to transfer some chemical functional group. So the transfer of functional group is a basic requirement for all the enzymes which are covered or listed under this category. So when take the example, it will be start with EC2 class, that is transferring one carbon group. If it is the case, then the number should be EC2.1. Then EC2.3, then acetyl transferases will be there. If we want to transfer, or if the enzyme is transferring nitrogenous group, then the number will be EC2.6. Likewise 2.7, 8 and all, they are transferring different type of the functional group like molybdenum and all. So that was all about the transferases class. Now the third class, that is hydrolases, right? So in this class, as we understand that the participation of hydrolysis occurs, right, and all this type of the enzymes will do the hydrolysis, that can be grouped together in this class which is known as EC3 hydrolases. If it is EC3.1, then it will be acting on the ester bonds. Then if it is in EC3.2, then they are glycosylases, and some will acted upon CN bonds and peptide bonds and halide bonds and phosphorus nitrogen bonds. So so on, it will be grouped and named like this, denoted by this EC3 point something, that is 1, 2, 3, depending upon the type of the bond which has been acted upon. So that is about that class. Fourth class, that is the group of enzymes which will go for Lyases, that is comes under the class that is known as Lyase. The examples we have tried to list it out, that is the EC4.1, that is for carbon-carbon Lyases. If the molecule which is acted upon will be making carbon-carbon position, it will have been acted upon by this position, then it is carbon-carbon Lyase, that is 4.1 EC. EC 4.2, that is carbon oxygen Lyases. If it is EC 4.4, then you can see the things that it is been acted upon by carbon sulfur Lyases. then if it is 4.99, then it is other Lyases. So that is the case. Now 5, that is isomer classes, Isomerases. As we have discussed also, that in this group the listing of the enzymes will be there, that the enzymes will act upon the substrate and convert that substrate into the product, which will be the isomer of the substrate. So depending upon which type of the isomers are created, which type of the reactions are going on, like on the basis of that, this type of the examples can be denoted. First one, as it is 5.1, that is racemase and epimerases. Then, as sir has discussed this particular example in detail, that is cis trans isomerases, that cis and trans positions can be gained by the action of the enzymes, that can be denoted as EC 5.2. 5.5, that is intramolecular lysis, and if it is EC 5.99, then some other isomerases can be there. So that is the group what we have discussed, that is isomerases. Now last group, that what we'll discuss, and we'll try to list out some examples for that. That is the last group is ligases. As we have discussed, that in this particular group, the enzymes which will do the function, that is of joining the bonds, right, between two substrates, that type of the enzymes will be listed upon. If the name, the number will be of enzyme commission 6.1, then they will make the bond forming between carbon and oxygen bonds. If it is 6.2, they will give carbon sulfur bonding. If it is 6.3, they will form carbon nitrogen bonding. If it is 6.5, they will give the phosphorus ester bonding. And if it is 6.6, then they will form the nitrogen metal type of the bonds. So these are some various examples of the exact names, that how this type of the naming of enzyme is been done, and after naming it, which type of the classification should be done. So as we all understand that classification and nomenclature both are interrelated, and without the idea of one, another cannot be understood properly. So for that, IUB has done the great work to give us a particular idea by only the number, right? And the name, as we discussed, there are two names for any type of enzyme. One is a trivial or a common name, another is a systematic name. But this idea to give the number denotation for each and every type of the enzyme will work very greatly